Hello students, welcome back to Vedantu NEET English. This is Baswaraj sir, your biology master teacher. So, before we start today's amazing class, that is photosynthesis in higher plants, line by line, every single word to word from NCERT, quickly let me know in the chat if I'm audible and visible to all of you. That's very important, right? See, if I keep blabbering here and later you tell me, sir, you're not audible, you're not visible, what is the point? So, quickly let me know in the chat right now if I'm audible to all of you. And this beautiful presentation, right? The beautiful sunset and the sunlight, which is important for the plants. The beautiful plants is visible to all of you. Quickly let me know in the chat, audible and visible. Yes. So students, with that being said, what are we going to do today in today's class, right? So obviously I'll be teaching you from the basics. We will be solving questions. Yes, we'll be solving a lots of PYQs of NEET and AIMS. NEET and AIMS PYQs. So for you to understand that, yes, whatever I've learned in Baswaraj sir's class, I can solve AIMS level question. That feeling you will get in today's class. Okay. In terms of content, we'll be learning the basics of photosynthesis, early experiments, what is a pigment, how do you extract a pigment. Then you will be learning something called as where does the photosynthesis takes place. Spoiler alert, chloroplast. Okay. Then we'll be also learning something called as light reaction, both cyclic and non-cyclic. So that is the entire journey for today's class. So let me know in the chat if you're excited for today's class with the help of NCERT line by line. Everyone, red hearts in the chat. Quickly, everyone, red hearts in the chat today. Red hearts in the chat. Now let me see how many of you can answer such simple questions. Look at this question. In light reaction, plastoquinone facilitates the transfer of electrons from. Now you might wonder, sir, in the NCERT, they have not mentioned such a term, plastoquinone. They have not mentioned cytochrome B6F complex. But how have they asked such question in NEET examination? As a matter of fact, in NEET 2006 also, there was a question which spoke about plastocyanine, iron sulfur proteins, cytochrome pyridoxine. Now you might wonder, sir, is this a little difficult? Students, is it a little difficult to answer these questions? No. Trust me, students, once you start learning it, once you learn from today's class, you will be able to solve every single question. Every single question you will be able to answer after today's class. Okay. So with that being said, let's start today's class, that is photosynthesis in higher plants. Can we start, students? Today's amazing, beautiful journey of today's class. 1 second, students. 1 second, let me just check if I'm audible and visible from my side also. Okay. Now, first of all, this is what I always teach all my students anywhere. What is the meaning of photosynthesis? What is the meaning of photosynthesis? Can anyone tell me in the chat? What is the meaning of photosynthesis? Photosynthesis. Obviously, you will tell me, sir, photo means light. Photo means light. And synthesis means to produce. Synthesis means to produce. That is, plants are taking light energy and they're converting into chemical energy. Light energy to chemical energy. Now tell me, students, what is the meaning of autotrophs? Anyone in the chat quickly? Autotrophs. Autotrophs are the organisms. Autotrophs are the organisms which can prepare their own food. Okay. So if I can make Maggie, for example, we have Abhyot, we have Pahul. Can Pahul make Maggie? He'll be like, yes, sir, I can make Maggie. So can I call Pahul as autotrophs? Quickly, yes or no in the chat. Or can I say, like, let's say Ravindrani. If Ravindrani can make, right, can make biryani, is it autotrophs? True or false? Quickly tell me in the chat. You're telling me autotrophs can prepare their own food, right? Is Rudranath an autotroph? Tell me. No. Autotrophs are the organisms which can prepare their own food. The meaning is that they can convert inorganic substances. They can convert inorganic substance into organic substances. Inorganic to organic substances. That is the meaning of that. For example, let me tell you a quick story. Not story, let me tell you a quick basic thing which I always tell. If this is a plant cell right here, tell me, do plants take in CO2? You will be like, yes sir, plants take in CO2. Okay. Now I told from inorganic to organic. In this inorganic components, can I include sunlight? Yes, include sunlight. The product or the food which I'm talking about is the glucose. Yes. So plants convert. All of you tell me, plants convert CO2 into glucose. Yes. Now, but if you look at the reaction here, CO2, carbon, oxygen and oxygen. Obviously, the missing link here is the hydrogen. Yes, hydrogen is missing. So if you have a garden in your house, all of you have garden in your house, right? If you have garden in your house, do you add water to the garden? So I'm adding water here. Okay. Now what exactly is happening here is this hydrogen right here is entering here. So what is happening here? Addition of hydrogen. Yes or no, quickly? Addition of hydrogen is happening. Addition of hydrogen. Now, if hydrogen is being added here, CO2 is getting converted to glucose. What is the fate of this oxygen here? Poor sad soul. Obviously, you tell me, sir, this oxygen right here will come out. O2 is released out. So, have you heard in your younger classes? You might have heard in your younger classes that plants take in CO2. Yes, they are taking in CO2 and plants release out oxygen. Yes, plants release out oxygen. All of you know this. Yes, all of you know this. Now, I'll teach you one more concept here. Addition of hydrogen. This is a very important concept to understand the entire chapter. Addition of hydrogen or electron. Removal of hydrogen or electron. Understand these two definitions. Addition of hydrogen or electron is called as reduction. It is called as reduction. While removal of hydrogen or electron is called as oxidation. It is called as oxidation. Now you tell me in the chat, if I am adding hydrogen here, that is right here students, if I am adding this hydrogen here, will this process be oxidation or reduction? Obviously, it is going to be addition of hydrogen is nothing but it is a reduction process. It is a reduction process. Clear all of you with me? It is a reduction process. It is a reduction process in which inorganic substances are converted to organic substances. Inorganic to organic substances. Clear? Now you tell me, if sunlight is here, sunlight is coming, yes, sunlight is falling. Now plant cells are absorbing the sunlight. Plant cells are absorbing the sunlight. Is this endothermic or exothermic then? Absorption of heat is called as endothermic reaction. It is a endothermic reaction. Endothermic reaction is happening here. Okay. Endothermic reaction is happening. Now can I also tell the CO2 is being reduced to glucose? Obviously, the CO2 is adding hydrogen and it is getting reduced to glucose. That is, CO2 is reduced. CO2 is reduced to glucose. C6H12O6. C6H12O6. That is, CO2 is reduced to glucose here. Done. Now, students, you might have heard about something called as metabolism. How many of you know what is metabolism? Now, you might have heard about in metabolism, there is something called as anabolism and catabolism. Right? What is anabolism? Anabolism is a process when you make something. From simple substances, we are making a complex substance. In catabolism, we are taking a complex substance and breaking it down. That is called as catabolism. Now you tell me, using simple substances, we are making a complex substance here. You tell me, is this reaction right here anabolic or catabolic then? Anabolic or catabolic? Quickly. So this reaction right here, where a complex substance is formed, can I call it an anabolic process? Yes. anabolic process. So this is about your, this is all the points which you need to know about your photosynthesis. Students, trust me on this. From this slide right here, behind my back, there are so many numericals or so many PYQs which has been asked. And whatever you learn in this slide is the foundation or the basics which you need to know about photosynthesis before you start the chapter. Okay, so with that being said, let's read the lines of NCERT and let's understand and decode every single line. Okay, let's decode. For example, this is the first paragraph right here. The first paragraph is right here. Okay, now all animals, including human beings, depend on plants for their food. Yes or no? Obviously, if I'm making Maggie, or if I'm making chapati in the house, the chapati has to come from the wheat has to come from the plants. So ultimately, any organism except your bacteria, they have to depend on plants. They have to depend on plants for food. Okay. Have you ever wondered from where do plants get their food? Have you ever wondered? Do plants, are they getting it from somewhere? No. They're preparing their own food. Plants are preparing their own food, right? Now, green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. So students, if there is a food chain, if there is a small food chain, in every food chain, especially grassland ecosystem, in a terrestrial ecosystem, every food chain, the primary producer is going to be a plant. True or false? True or false quickly? Yes. So if the primary producer is a plant, can I tell every other organism in that food chain has to depend on the plants for the food? That's what they're saying here. A green plants make or rather synthesize the food they need through photosynthesis. And are therefore called as autotrophs. They're called as autotrophs. Plants which can make their own food. Primary producers. Okay. Now, next. You have already learned that autotrophic nutrition is found only in the plants, and all the organisms that depend on the green plants for the food are called as heterotrophs. So can I call Ovia? Can I call Hari? Can I call Rashmita as heterotrophs? Yes. Including me, all of you are heterotrophs. Now, green plants carry out photosynthesis, a physico-chemical process. It is a physico-chemical process. Now, you might ask me, sir, what is so physical and what is so chemical about it? Let me tell you. Before we go, let me read the paragraph. By which they use light energy to drive the synthesis of organic compounds. Okay. Now, you might ask me, sir, what is the difference between this physical process and chemical process? What is this? Physico-chemical. Sounds very fancy, but it is very simple. Okay. Now, first understand the physical process. Okay. Now, students, there is absorption of light here. Tell me, is there absorption of light when it comes to photosynthesis? Yes. So, can I tell the first point here? Can I tell absorption of light energy? Absorption of light energy. Absorption of light energy. Okay. Then absorption of light energy. Yes. Exchange of gases. Yes. Plants are taking in CO2, releasing out oxygen. Exchange of gases is a physical process. Yes, it is a physical process. Then, when the light energy is falling on a pigment, from the pigment's outer shell, can electrons get excited? Yes, all of you in the basic chemistry. So, excitation of electrons. Excitation of electrons. Excitation of electrons. All of the following are a type of physical process. Physical process. Now, what about chemical process, students? Chemical process. What are the chemical process here now? Formation of bonds. Formation of bonds. For example, your carbohydrate is getting converted to C6H12O6. There is bond formation here. Yes, there is bond formation. Bond formation is there. Now, tell me, in the first slide I told you, right here, the water is providing the hydrogen, oxygen is being released out. Can this happen without the breaking the bond here? Obviously not. So can I tell photolysis of water? Photolysis of water. Yes. Photolysis of water is happening. That is breaking of bonds. Breaking of bond is happening. Then also conversion of energy is happening. Yes. Conversion of energy, that is light energy, is getting converted to chemical energy. Chemical energy is getting converted. Yes, that also is a chemical process. Students, you need to understand your entire photosynthesis is a physico-chemical process in which there are physical factors and there are chemical factors which are working together to make the food for the plants. Okay, not for us, for the plants themselves. Okay. Now, next paragraph. Ultimately, all living forms on earth depend on the sunlight for the energy, right? They depend on the sunlight. Not all the organisms. There are certain bacteria which depend on organic compounds, that is the chemical compounds. The use of energy in the form of sunlight by plants doing photosynthesis is base of life on the earth. True. Photosynthesis is important due to the two main reasons. Again, very important lines now. Very important lines. I want everyone to highlight here. What are the two main importance? What are the two main importance for the photosynthesis? Two main important things. First one, okay. First one is given here. It is the primary source for all the food on earth. Yes, that is, plants are primary producers. First point, done. It is also responsible for the release of oxygen into the atmosphere by the green plants. So the first point here is what? The first point here is primary producers, plants, and the second point here is plants are releasing oxygen as a byproduct. Here we learned here, right? See here, we learned here plants are releasing oxygen as a byproduct, which is useful for the humans, which is useful for our humans. Okay, that is the meaning of the two main important things. Okay, now this chapter focuses on the structure of photosynthetic machinery and various reactions of that transforms light energy into chemical energy. Okay, so with that, the starting basics of the chapter you know now. All of you are aware of the basics of the chapter now. Clear. Now we learn something called as experiments now. Early experiments to prove that, you know, plants require CO2, plants require sunlight, plants require chlorophyll, plants require water, plants are producing glucose. All of these experiments we'll learn. For example, if you go back here again, this slide, let me show you the importance of this slide. If you go back to this slide, what we'll be learning right now is, we'll be showing you how plants require CO2 for photosynthesis. We'll be learning plants require water for photosynthesis. We'll be learning that oxygen is the byproduct. We'll also be learning that glucose is stored in the form of starch with the help of different types of experiments, one by one. Till now here, any doubt, quickly ask me in the chat. Any doubt till now? Quickly, all of you. No doubt? If there is no doubt, let's start with the experiments. Let us try to find out what we already know. Then why are we learning then? If NCERT is telling, we will have to learn something which we already know now. Okay. about photosynthesis. Some simple experiments you have done in the earlier classes that shown that chlorophyll, green pigment of the leaf, light, CO2 are required for photosynthesis to occur. Without chlorophyll or without light or without CO2, photosynthesis cannot happen at all. Photosynthesis cannot happen at all. Okay. Now, the first experiment is called as a variegated leaf experiment. The first experiment is called as what? Variegated leaf experiment. Now, what is the meaning of variegated leaf? The meaning of variegated leaf is that it has variable amount of chlorophyll. See, if you look at this leaf right here, you can clearly tell me that in certain regions there is very high chlorophyll. You know, see the dark color regions, very high chlorophyll here, very high chlorophyll here. In the dark regions, very high chlorophyll, and in the lighter regions there is very less chlorophyll is there, very less or no chlorophyll. Such a leaf which has variable amount of chlorophyll, some places very high chlorophyll, some places very low chlorophyll, that is called as variegated leaf. That is called as variegated leaf. Okay. Now students, to this variegated leaf I am adding sunlight. Okay, sunlight. Now what happens? After some time you take the plant and you take the leaf out. To this leaf you need to add iodine. You need to add iodine to it. Now once you add iodine to this leaf, wherever we had the dark green regions, the dark green regions are converted to blue-black. Can you see? They are converted to blue-black. And wherever we had the lighter regions, it will remain as such. Now, what is the conclusion here? The conclusion is very simple. Wherever we had chlorophyll, wherever chlorophyll was present, photosynthesis took place. If photosynthesis took place, there was production of starch. If the starch was present, iodine acted on this starch and it converted the starch into blue-black color. What about in the lighter regions? In the lighter regions, there was no chlorophyll and there is no blue-black formation. This simple experiment shows you that for a photosynthesis to take place, for a photosynthesis to occur, plant need chlorophyll. No chlorophyll, no party. Okay, simple. No chlorophyll, no party at all. Okay, friends, if you are able to understand such simple experiments, quickly smash that like button, all of you, and mark your attendance, all of you, quickly. Even if you are watching it recorded, quickly smash that like button. Okay, now you have carried out the experiment to look for starch formation in two leaves. A variegated leaf, a leaf that has partially covered with black paper and exposed to light. So basically it is two experiments are there. One is variegated leaf. The other leaf is very simple. The other leaf, you take a leaf. This is a leaf, right? In this leaf, you need to cover this part with a black paper. This part, one side with a black paper completely. One side with a black paper, other side of it is normal. So wherever you have covered with a black paper, there will be no photosynthesis. Here there is no black paper. Here there is no black paper. So photosynthesis will take place. Same principle again. Same principle. Okay. Now, on testing these leaves for a presence of starch, it was clear that photosynthesis occurred only in the green part of the leaf in the presence of light. So whenever there was green leaf was there, it is present. Very simple experiment. If experiment is clear to all of you, quickly, green has no chat if it's clear to all of you. I want to know the confirmation. No. Now, next experiment. Now, next experiment is called as Moll's half leaf experiment, also called as KOH experiment. KOH experiment. Very simple again. Very simple experiment. Okay. Now, if you look at this experiment here, half-leaf experiment, if you can notice, half of the leaf is inside a jar and half of the leaf is outside in the environment. Two simple parts. Okay, half leaf is inside the jar, half leaf is outside. Okay. Now, what is so special about this jar? The speciality of this jar is because inside the jar there is a chemical substance called as KOH. Inside the jar right here, there is a chemical substance called as KOH, potassium hydroxide. Now, what this KOH does is, this KOH is very cunning. This KOH will completely remove all the CO2 inside. So, what is happening here? Inside the jar, there is absolutely no CO2. No CO2 at all. This KOH will completely absorb all the CO2. Now, you expose to sunlight. Okay, exposed to sunlight. Now, after some time, you take the leaf and again add iodine. Okay. Once you add iodine again, what is happening? The leaf which was covered in the jar, part of the leaf which is covered in the jar, there is no starch formation. Why? Because in this region right here, there was no CO2. If there is no CO2, it will never get converted to glucose. If there is no CO2, there is no conversion. If there is no glucose, obviously no starch. In the above region, which is exposed to the air, in the above region there is starch formation. Why? Because in the above region there was CO2 was present. So this simple experiment proves that. The simple experiment of half-leaf or Moll's half-leaf experiment proves that CO2 is very important for photosynthesis. No CO2, no party. Simple. Understood all of you? So carbon dioxide is very important for photosynthesis. Carbon dioxide is very important for photosynthesis. That's all. Let's see what is given here. Another experiment you have carried out where a part of the leaf is enclosed in a test tube containing KOH, soaked cotton, which absorbs the CO2. So KOH will completely absorb all the CO2. Okay. While the other half leaf is exposed to the air. Done. The setup is then placed in light for some time on testing for the presence of the starch. Later in the two part of the leaf, you must have found that the exposed part of the leaf tested positive for starch. Obviously, the part of the leaf which is exposed to the air is positive for starch. This one right here. While the portion that was in the tube tested negative. Why? Because this shows that the CO2 is required for photosynthesis, and if there is no CO2, there is no photosynthesis. If you understood the second experiment, quickly give me in the chat. I'll know that you understood the second experiment, all of you. Okay, understood the second experiment. So KOH is absorbing the CO2. Done. Now, students, the next experiment is called as bell jar experiment. It is called as bell jar experiment. Very simple again. Very simple experiment. Now, in bell jar experiment, what are we doing is, not we, Joseph Priestley uncle. Joseph Priestley uncle is doing what here? He is taking a bell jar. This is a bell jar. Inside this bell jar right here, there is a mouse, there is a candle. There is a mouse, there is a candle. Now we are killing the mouse. How? Suffocation. This burning of candle and this mouse right here or mice right here will respiration and combustion consume oxygen? Yes sir, quickly in the chat. Does the burning of candle, which is combustion, and this mouse right here is doing respiration, will it consume oxygen? Yes. Once all the oxygen inside the bell jar, it is consumed completely, will the mouse die? Will the candle become extinguished? Obviously. So after some time, what happens? After some time, the mouse is dying and the candle is also extinguishing. So Joseph Priestley thought, something is present in the air. He's like, something is present in the air. While breathing or respiration or combustion is damaging it. So he told that burning of candle and respiration by the mouse, it is damaging the air. And we know what is the damage. The damage is nothing but the oxygen in the air is being completely consumed. That is what he meant by damaging the air. Later on, what did he do? Later on, he added a mint plant inside. So obviously, if the mint plant is added, combustion and respiration, if it is consuming the oxygen, will the oxygen be replenished by the mint plant? Obviously, this mint plant will release more oxygen. And this particular cute mice and this candle will keep on burning. Yes. So see here, it is burning here. So he later told us that. So what we understood now, students? It is interesting to learn. Okay, here. Joseph Priestley in 1770, very important date, performed a series of experiments that revealed the essential role of air in the growth of green plants. So what is important now? Air. What exactly is in the air? Oxygen in the air is very important. Oxygen air is very important. Priestley, right? You may recall, discovered the oxygen. Very important line. He discovered the oxygen in 1774. Okay. Priestley observed that a candle burning, combustion, in a closed space, a bell jar, soon gets extinguished. That is, candle is going. Okay. Similarly, a mouse would soon suffocate in a closed space. He concluded. What he concluded? He concluded that a burning candle or an animal that is breathing the air somehow damage the air. They somehow damage the air. What is the meaning of damaging the air here, students? Everyone in the chat? The meaning of damaging the air here is the oxygen is being completely consumed by the mouse or the plant. Mouse or the plant is completely consuming the oxygen here, right? But when he placed the mint plant, I told you, mint plant, they can ask you a question on this. Mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. So tell me, what do you understand from here, students? Tell me, what do you understand here? From this experiment right here, it is understood that for a photosynthesis to take place, oxygen is important. Then oxygen is CO2 is very important. Not oxygen, CO2 is very important. And with the help of photosynthesis, byproduct is oxygen. Byproduct is oxygen. Release of O2 by mint plant. Done. Understood? CO2 is necessary for the plants and oxygen is the byproduct of the photosynthesis. Oops, the mic only fell off. Lol. Okay, done. What is he hypothesized? He hypothesized that, he told us that plants restore, very important line, plants restore to the air whatever breathing of animals and burning of candle removed. So breathing of animals and burning of candle completely removed the oxygen and plants restore the oxygen. That is, plants release the oxygen. Third experiment, then understand. Smooth, right? All of you understanding in the chat quickly? Tell me in the chat. Students, all of these are very important because questions have come from here. It has come in your examination. I'll show you. Right? No. Next. Now there is one scientist called as Ingenhousz. Ingenhousz scientist. He copied the same experimental setup of Joseph Priestley and he did some slight modification. What is the modification now? Let's see. This is Joseph Priestley's. This is the experimental setup of Jan Ingenhousz. Now Jan Ingenhousz told us that when you take a particular plant in a water or any other medium also, right? Any other normal land also, plant is also fine. When you take a plant, it is able to produce bubbles. Obviously here it is bubbles are indicating oxygen here. Bubbles are indicating oxygen because it's a byproduct of photosynthesis. This bubble will release only in the daytime. Only in the daytime. But if you take the same plant and you keep it in shade or night time, there are no bubbles here. What is the experimental, what do you get out of it now? Tell me. The friend who gets more marks, all of you. Exactly, Kanmani. The friend who gets more marks. What do you call this? This is basically telling you that sunlight is important. That's all. So it is, if there is sunlight, there is photosynthesis. If there is no sunlight, no photosynthesis. All of you understood? All of you understood, right? So if there is sunlight, photosynthesis will happen. Obviously, the proof here is bubbles. So bubbles are basically oxygen, and we know oxygen is the byproduct of your photosynthesis. Understood? This is the experiment given by Jan Ingenhousz. Jan Ingenhousz gave this experiment. Let's read what is given here. Let's read what is given here. See, using a similar setup as one used by Priestley, told you, is copying the same experimental setup by Priestley, Jan Ingenhousz. Okay, but only by placing it once in dark and once in sunlight. See, he's copying the is using the same setup as Priestley, but new theory. He also became famous. Okay, Ingenhousz. Jan Ingenhousz showed that sunlight is essential to the plant's process that somehow purify the air fouled by the burning of candles or breathing of the animals. Ingenhousz in an elegant, elegant experiment. See, elegant experiment. Okay. Elegant experiment with an aquatic plant showed that in bright light, small bubbles were formed around the green plants, while in the dark, they did not. So in the dark, there was no release of oxygen. Okay. Later, he identified these bubbles to be the oxygen. Okay, they were oxygen. Hence, he showed that it is only the green parts of the plants that could release the oxygen. You did not get release of oxygen from the roots and all. Remember that. From the roots, there is no oxygen. Very simple. Okay. This is the experiment given by Jan Ingenhousz. Okay. Next one. Next one is by Julius von Sachs. Weird name, I'm telling you again. In the year 1854, Julius von Sachs provided the evidence, what did he tell us, for the production of glucose when the plants grow. So what he tell us was, let me tell you some points here. Let me tell you points, what did he tell? First point here. Glucose is usually stored in the form of starch. First experiment. First thing he told. Second thing he told was what? Green substance in the plant, chlorophyll, as we know it now, is located in a special body called as chloroplast. That is the second one. So first one is what? Glucose is stored in the form of starch. Second one is chlorophyll is present in the chloroplast. Chloroplast. Okay. Third one. He found that the green parts in plant is where the glucose is made. That's all. And glucose is stored in the form of starch. So wherever we have green leaf, you will find glucose there initially. 3 points, done. Done? Done all of you? 3 different points for Julius von Sachs. First point is what? Glucose is stored in the form of starch. Second point is that green substance is called as chlorophyll and you find the chlorophyll in the chloroplast. Third point is that in the green parts we find glucose is made. That's all. 3 points, done and dusted. Now, next experiment. Next experiment is given by T.W. Engelmann. T.W. Engelmann. Okay, where is his name? Let me highlight his name. T.W. Engelmann or Engelmann. Yeah, Engelmann. It sounds like Eagle Man, or you can call it as Engelmann. Angle or Eagle Man. Okay. Very simple experiment. Understand the experiment. Okay. Here we have a prism. Yes, this is a prism right here. Okay. We have a prism right here. We have a white light coming in, and light is scattering now. Light is scattering. Yes, light is scattering. Okay. Now what did he do was very simple experiment. Very simple experiment. he took a test tube here. Yes. He took a test tube. He took a test tube. Now, inside this test tube, students, inside the test tube, he added an algae. He added an algae is inside. A filamentous algae. Okay. A filamentous algae is inside. Filamentous algae is on the inside. Okay. Now, what is happening here? This light right here is falling on the algae. Falling on the algae. Which light? All types of light. All the way from the red to blue. All the way from the red to blue, all the different 𝐂𝐤 𝐎𝐤 is falling. Okay. Okay, 𝐂𝐤 light is falling on the entire algae. This algae right here is called as cladophora, which I'll tell you later. I'll show you in the paragraph later on. Cladophora. Now, students, in this test tube, which was filled with water, it is filled with water. In this test tube, which is filled with water, there were certain bacteria. There were certain bacteria called as aerobic bacteria. And this aerobic bacteria love oxygen. Aerobic. They love oxygen. Okay. So, what do we have here? We have the aerobic bacteria inside. Aerobic bacteria on the inside. Aerobic bacteria on the inside. Now, after some time, Jan Ingenhousz noticed that. After some time, Jan Ingenhousz noticed that this aerobic bacteria is clustering, or it is forming one gang, one nice gang it is making in these two different regions. This particular aerobic bacteria is forming a gang around here. This aerobic bacteria is forming a gang here. The same aerobic bacteria is forming a gang here in these two regions. In these two regions, aerobic bacteria is forming a gang. Now you might ask me, sir, why are they completely making a gang there? Tell me. In your class, if someone brings a nice biryani to the class, will all of you go there or not? So, for example, let's take someone from the chat, Madhumati, she loves biryani. Okay. If he doesn't like also, for the video you will like now. Okay. In a class, if someone is bringing a biryani, will you not go with the smell only? You will take the smell and you will go there. Yes, you will go there. The same thing as bacteria is doing. Since this bacteria loves the oxygen, it is clustering around here. So can I tell in this region right here, there is more aerobic bacteria. Aerobic bacteria is more here. Yes, aerobic bacteria is more here. In this region right here, can I tell there is obviously more oxygen is here? Yes, more oxygen here. If there is more oxygen, which is the byproduct of your photosynthesis, can I tell in this region right here, there is more photosynthesis? There is more photosynthesis here. Yes, more photosynthesis here. So what does it tell you, students? What does it tell you? It tells you that in these two regions right here, that is the red region and the blue region, the red region and the blue region is biryani is found here. Meaning is that in this red region and the blue region is the favorite wavelength of light. I repeat again, red region, the blue region is the favorite wavelength of light for your plants. Okay, so in this red region and the blue region, since it is very favorite for the plants, this particular algae is able to do more photosynthesis in the red region and the blue region. In the red region, the blue region, the plant is able to do more photosynthesis. Understood, all of you? So, can I tell some work is done here now? Some work is done. Plants are doing some work. If you are not doing some work, plants are doing some work. What is the work here? Work here is photosynthesis. So, can I tell photosynthesis is happening here? So, what does it tell you, friends? It tells you something called as action spectrum. We'll get to that later on. Action spectrum. What is the action spectrum? Action spectrum is nothing but at which wavelength photosynthesis is happening. at which wavelength, right up, easy for everyone to see, at which wavelength photosynthesis is happening. So can I tell in this red region and this blue region, photosynthesis is happening more? Yes, happening more. Students, you need to remember Jan Ingenhousz, T.W. Engelmann. T.W. Engelmann gave the first action spectrum also. He gave the first action spectrum. Action spectrum is a spectrum which tells you at which wavelength photosynthesis is happening. That's all. We'll get to that later on again. Okay. Let's read what is given here now, students. For you to understand better, I'm giving you NCERT. Okay. T.W. Engelmann. Using a prism, he split light into spectral components. Yes. Then illuminated on a green algae, that is Cladophora, placed in a suspension of aerobic bacteria. Okay. The bacteria were used to detect the site of O2 evolution. He observed that bacteria accumulated mainly in the regions of blue and red regions. They accumulated completely. Of the split spectrum, the first action spectrum of photosynthesis was thus described. First action spectrum. It resembles roughly the absorption spectrum of chlorophyll A and B, which we'll discuss later on. What is absorption spectrum? I'll tell you later on. Very simple again. Very simple absorption spectrum also is. Okay. Any doubt in this paragraph? I believe there is not. Okay. Now, students, let's understand what we learned till now. What have we learned till now? Okay. By the middle of the 19th century, the key features of the plant photosynthesis were known. Namely, the plants could use the light energy. All of you know Jan Ingenhousz. Yes. To make carbohydrates from CO2 carbohydrates, half-leaf experiment. And water is very important, hydrogen donor. Yes. The empirical equation for representing the total process of photosynthesis for oxygen evolution organism was then understood. CO2 plus water, light energy, and in the presence of chlorophyll, we get C6H12O6. That is glucose. And oxygen is the byproduct here. Now, students, in this, all of these biologists and all of this botanists and zoologists, there was a microbiologist. Microbiologist should stick to microbes, but he made an interesting observation. This microbiologist observed that in the case of bacteria and in the case of plants, the hydrogen donor are very different. For example, students, I told you in the case of plants, the hydrogen donor, is water. That is, the entire reduction process is because of water is splitting, and the hydrogen is given here, and the oxygen is the byproduct here. But in the case of certain bacteria, students, that is not the case. That is not the case in the case of certain bacteria. For example, in the case of green and purple, sulfur bacteria, your hydrogen, that is water, is not the hydrogen donor. For example, in such bacteria, there is no water. Water is not the hydrogen donor. So if water is not the hydrogen donor, obviously oxygen will never come out. So what exactly is happening here? That is, they did not release oxygen during photosynthesis. So such bacteria, green and purple sulfur bacteria, use for hydrogen donor. They use hydrogen donor as H2S, hydrogen sulfide. They use hydrogen sulfide. Now you tell me, if this hydrogen right here, right, students, if this hydrogen right here is given to this CO2, to make glucose, what will be the byproduct now? Obviously, in this case, in the above case, what happened? In the above case, which in the case of plants, this hydrogen was given here and oxygen was the byproduct in the case of plants. But in the case of this green and purple sulfur bacteria, in the case of this green and purple sulfur bacteria, the byproduct is going to be sulfur. The byproduct is going to be sulfur, and oxygen is not released here. Oxygen is not released here. So the hydrogen donor is, in the case of this bacteria, is H2S, and in the case of green plants it is H2O. It is H2O. All of you understood? That is called as this entire work was done by a microbiologist. This entire work was done by a microbiologist. His name is Cornelis van Niel. Cornelis van Niel was a microbiologist. Can you see here? Who, based on his studies on purple and green bacteria, demonstrated that the photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces the carbohydrates. That is, water is giving the hydrogen to CO2. CO2 is being reduced. Carbon dioxide is reduced to carbohydrate. Okay. Now in green plants, H2O is the hydrogen donor and it is oxidized to, obviously glucose is removed out. Some organisms do not release O2 during photosynthesis, which when H2S, can you see here, hydrogen sulfide, instead is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur here, which is the byproduct. Sulfur is coming out, or sulfate. Okay, on the organism and not O2. Hence, he inferred that O2 evolved by the green plants comes from water. The oxygen is coming out. All of you know, right? All of you know that when the plants do photosynthesis, oxygen is the byproduct, and this byproduct is coming from water. This byproduct is coming from water and not from the CO2. It is coming from this oxygen right here. O is coming from water. This O is not coming from CO2. Why? Because CO2 is getting converted to glucose. CO2 is getting converted to glucose. That's all was his hypothesis. Can you see? And not from carbohydrate. Okay? Not from carbohydrate. Clear? This was later proved by using the radioactive isotope techniques. That is, instead of using 16 oxygen, they used 18 mass, higher isotope of oxygen, and they tracked it. You know, where is the oxygen going? I've given a simple example before, right? The simple example was you have a cat in your house. To the cat, you're adding a GPS, and then you're tracking where the cat is going. It's going to jungle, wherever it is going. Is the cat going to neighbor house to drink more milk? You will know if you add a GPS in the cat collar. Similarly, to the oxygen, we're adding a tracker here. The tracker here is radioactive isotope. So whenever the oxygen travels anywhere, you're able to track it. Okay. The correct equation that would represent the overall process of photosynthesis is therefore right here. 6 CO2 plus 12 H2O. It is balanced equation, right? Balanced equation. 6 CO2, 12 H2O in the presence of light and chlorophyll. And chlorophyll, we get glucose, water, and oxygen as the byproduct. O2 is released in the form of water. O2 is released in the form, is from water. This was proved by radioactive isotope technique. Okay. Note that this is not a single reaction, but a description of multi-step process called as photosynthesis. And remember, students, photosynthesis is not a single step, it is a multiple step reaction. There is light reaction, there is dark reaction. All that is happening in the photosynthesis. Students, are you getting that feel of the chapter now? All the different reactions, all the different types of scientists who worked towards the discovery of how the plants work, the mystery behind how the plants are able to produce the food. The mystery is what we're going to solve right now. Ready? So repeat the star. What star? What star? There's no star here. Okay. So Cornelius Van Til told us what? Cornelius Van Til told us that the hydrogen donor is water in the case of plants, and in the case of bacteria it is H2S. Byproduct is water, which is coming from oxygen is coming from water. Okay. Now students, with that being said, the next thing which we need to understand right now, is where does the photosynthesis take place? Where does the photosynthesis takes place? Okay. Now, if I ask all of you, or if I ask a class 10 student, for example, if I go on the road and I ask a class 10 student or class 8 student, where does the photosynthesis takes place? Tell me. What will the student tell? The student will say, sir, photosynthesis happens in the green plants, right? He will tell photosynthesis happens in the green plants. I'll be like, okay, sir, green plants. Now, in the green plants, specifically, can I tell in the leaf? Yes, in the leaf. And in the leaf, can I tell specifically in the mesophyll cells? Mesophyll cells. In the mesophyll cells, can I tell specifically the chloroplast? Chloroplast. Yes, in the chloroplast. Specifically inside chloroplast, where does it happen? That we will learn now. Okay. So if someone asks you a question, where does the photosynthesis takes place? Please don't tell leaf, tell chloroplast. At least tell chloroplast. Okay. So photosynthesis takes place inside the chloroplast. Inside the chloroplast. Now let's draw the chloroplast with me, all of you. Draw the chloroplast with me now. All of you quickly draw the chloroplast with me now. Chloroplast is a double membrane structure, right? Chloroplast is a double membrane structure. So first membrane, second membrane. Done. Double membrane structure. Chloroplast is a double membrane structure. Double membrane structure. So can I tell students, here we have the outer membrane, and we have the inner membrane. Outer membrane as well as the inner membrane. Two different membranes are here. Outer membrane as well as the inner membrane. Now, what do we have inside, students? What do we have in the inside? If you look at the insides of a particular chloroplast, you will find certain disc-like structures. You will find certain disc-like structures like this. Okay. You will find certain disc-like structures. This disc-like structures are called as thylakoids. This disc-like structure, you see, this disc-like structures are called as thylakoid. Thylakoid. Okay, thylakoid. Clear? Thylakoid. Now, this single disc is called as thylakoid. Now, what about this entire stack? This entire stack is called as grana. Grana or granum. Grana or granum. This entire stack is called as grana or granum, entire stack. Okay. Now here, you need to understand one more thing. All of this grana are connected. All of this grana are connected to each other. Can you see? All the grana are connected to each other. All the grana are connected to each other. The connection is called as stromal lamella. Can you see this connection? This connection right here is called as stromal lamella. They're called as stromal lamella. Okay. Now, in this particular structure of chloroplast, you find the matrix also. Obviously, there will be a matrix here. Can you see, students? This is the matrix. We have the matrix here. Matrix. Okay. So we have a matrix here. The matrix, the gel-like structure, the matrix, like how we have chloroplast, how we have the cytoplasm, right, in the cell. Similar to the cytoplasm, we have the matrix here. The matrix is called as stroma. The matrix is called as stroma. Okay. Matrix is called as stroma. Matrix is called as stroma. Okay. Now, students, I want you to understand a few things here, which is very important for you to understand. Question will directly come like that. Okay. Now, students, on this thylakoid surface, we find two different, on the thylakoid structure, use different color so all of you will understand. On this thylakoid structure, we find two different types of photosystems. On this thylakoid, which you have right here, we find PS2 and we also find PS1, not PlayStation, photosystem 2 and photosystem 1, not PlayStation. Okay, photosystem 2 and photosystem 1 both. Now, what about the stromal lamellae? On the stromal lamellae, we find only and only, on the stromal lamellae, we find only and only PS1. PS1. So PS1 and PS2 both, PS1 and PS2 both are present on the thylakoid, while on the stromal lamella or lemma, stromal lamella, we find only and only PS1. We find only and only PS1. Now, write down one of the most important things right now. Okay. Now, let me tell you something. Where does the light reaction and where does the dark reaction happens? Pauline Salvino, PS1 movie are going all of you. Now, understand. Okay. Understand that where does light reaction happens? Light reaction happens on the following structures. Light reaction. Don't worry what is light reaction, I'll teach you later on. What is light reaction? Light reaction takes place on the stromal lamella, on the entire thylakoid. There we have the light reaction. Then where does the dark reaction takes place? Okay, dark reaction takes place in the matrix. Can you see? Here the dark reaction takes place. It doesn't mean it will happen on the dark side. Don't tell you that. Here we have the dark reaction taking place. Dark reaction taking place here. Okay, dark reaction. Now, students, tell me, do the particular structure of chloroplast, do they have their own DNA? Yes, they have their own DNA. They will have their own DNA. Okay, they will have their own DNA. They have their own DNA. Now, tell me, will they have their own ribosomes? Yes. Will they have their own ribosomes? Yes, they will have their own ribosomes. 70 years. Ribosomes can be found here, just like a bacteria. Just like a bacteria. This is the structure of a chloroplast. This is the structure of a chloroplast. Structure of a chloroplast. Done. 70th ribosome is present. Okay. Now, students, with this being said, let's see what is given you NCERT. Let's read what is given in your NCERT now. Okay, you would recollect from the previous unit, that is mesophyll cells in the leaves have a large number of chloroplasts. They have a large number of chloroplasts. Previous chapter which they are talking about here is anatomy of flowering plants. Students, in the anatomy of flowering plants, have you done the cross section of a leaf? All my DNA students also tell me. DNA RNA students tell me, in the anatomy of flowering plants, have you done a cross section of dicot leaf and monocot leaf? Yes, we have done cross section of a mesophyll cell, the palisade mesophyll, especially palisade as well as palisade as well as your spongy parenchyma, spongy mesophyll you have done. If you remember, students, if you remember, on this palisade mesophyll, the middle part we had a large vacuole. We had a large vacuole in the middle, and all the chloroplast, students, all the chloroplast was always found in the periphery. All the chloroplast was found on the periphery. Like this. All the chloroplast was found on the periphery here. Okay. Usually the chloroplast align themselves along the walls of the mesophyll cells. Yes, they align themselves on the walls of the mesophyll cell such that they get the optimum quantity of incident light. Obviously, students, use logic. If the light is coming, if they hide inside, will they get light? Tell me. If they hide inside, will they get the light? No, obviously not. They will not get the light. The only way they can get light is all the chloroplasts are arranged on the walls. Okay. Here? Here they are telling you something extra here. Basically, when will they be perpendicular, when will they be parallel? They will be perpendicular when the light is very low, parallel when the light is not present. That's all. Okay. Now, students, let's see what is given in NCERT now. You have studied the structure of chloroplast in the chapter 8, right? Chapter 8, that is your cell unit of life. Okay. With the chloroplast, there is a membrane system consisting of grana, the stroma lamellae, and matrix, the stroma. Okay. Now, there is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also synthesis of ATP and NADPH. This is basically the stroma lamellae and thylakoid. The stroma enzymatic reaction synthesizes sugar. The dark reaction in the stroma, which is true, which turn form the starch later on. The former reaction, since they directly light driven, is called as the light reaction. What is the meaning of light reaction, students? The meaning of light reaction is that they are able to trap the sunlight and produce ATP and NADPH. Here, produce NADPH and ATP. Trap the light energy. That is called as light reaction. Also called as photochemical reaction. Very important. Very important. Because in the examination, sometimes they will not give you light reaction. Examination, they will never give you light reaction. They will tell you photochemical reaction of the photosynthesis. That time you should tell me, sir, light reaction is also called as photochemical reaction. Photochemical reaction. Okay. The later, basically, what is later? Later here they are talking about is the enzymatic reaction in the stroma. The later are not directly light driven, but are dependent on the products of the light reaction. Understand? I will clear one confusion for all of you. You cannot tell, later on to me, the light reaction happens in the light, dark reaction happens in the darkness. No. It doesn't work like that. The main difference is that light reaction needs light. Dark reaction does not need light. That is the main difference. Dark reaction does not happen in darkness. Both light reaction and dark reaction happen in the daytime only. Clear that doubt, all of you, from your mind. Both light reaction and dark reaction, both of them will happen in the daytime only. It's just that dark reaction does not need light. It does not need light, but it needs products of light reaction. Understand? That is, light reaction is giving you ATP and NADPH. Light reaction is giving you ATP and NADPH. The dark reaction is consuming that. Darkness, no work on its own. Dark reaction will consume the ATP, will consume the NADPH and do its work. Understood, all of you? So can I call dark reaction? Can I call dark reaction as light independent reaction? Can I call dark reaction as light independent reaction? Because they don't require light. Understood, all of you? Very simple concept, right? Why am I telling this so stressful is because in the examination they'll ask you a question. Which of the following is true about light independent reaction? Question will be asked like this. They'll never tell you dark and light. They'll tell you which of the following statements are true for light independent reaction. That time quickly in your mind you should tell they're asking about dark reaction. Okay? They're asking you about dark reaction. Clear? The dark reaction is also called as carbon reaction. However, this should not be constructed to mean that they occur in darkness or they are not light dependent. Okay? Understood, all of you? Very simple. Understand the points. If this paragraph is understood, all of you, green as in the chat. This is the structure of chloroplast. Outer membrane, inner membrane, stroma, lemma, grana, stroma, ribosomes, starch granules, lipid droplets. These are the storage materials. Now, with that being said, now we will go to the next concept for today. Today's class, that is the pigment. Tell me how many of you know what is a pigment? How many of you know what is a pigment? What is a pigment? Tell me. What is a pigment? What exactly is a pigment now? What exactly is a pigment? I'll give you 10 seconds to answer. Don't tell me, Madhu, just tell me examples of pigment. Tell me what is a pigment actually. All of you drink some water. So all of you will tell me, sir, color. Pigment means color. Okay. That is true. Pigment provides color. But can I tell this pigment right here? Is a chemical substance? It is a chemical substance. Yes, pigment right here is a chemical substance or a chemical compound. Right, it is a chemical substance compound which converts light energy into chemical energy. Which can convert light energy into chemical energy. Not understanding? Let me teach you. Okay, understand. If we have a pigment right here, understand the simple definition of a pigment. We'll do it here. Okay, we'll do it here. Now, I have a pigment right here. Okay, I have a pigment here. On this pigment, the sunlight is falling. Yes, on this pigment right here, sunlight is falling at a fixed wavelength. At a fixed wavelength, sunlight is falling on this pigment right here. Okay. So as soon as the sunlight falls on the pigment, the electrons in the outer orbital gets excited. Yes. Electrons in the outer orbitals are getting excited here. Yes. It is getting excited. Electrons are getting excited. Now tell me, if an electron is getting excited, will it come back to ground state? Yes, it will come back to ground state. This electron right here, eventually it will come back to the ground state. Okay. So when the electron is coming back to the ground state, there are multiple things which can happen. When the electron is coming back to the ground state from the excited state, couple of things can happen. For example, this particular electron can release heat, it can release light, it can release light, yes, it can release heat, it can release light, yes. The third thing is that this particular energy right here, can it be transferred to another pigment? Transfer of energy to another pigment, transfer of energy to another pigment. Okay. One more thing is that plants can use this energy to make some chemical reaction. They can use the energy use for chemical reaction. That is called as photochemical reaction. Out of these four things, students, the first two we do not want to learn. That you will learn in physics and chemistry. These two things, transfer of energy and photochemical reaction, these two things we will learn in today's class. I will teach you in-depth concept here. No mugging up in my class. Okay, in-depth concept. That is transfer of energy to another pigment. That is auxiliary pigments providing energy to the reaction center. You learn that. Use of energy to do photochemical reaction. That is ATP is being directly used in the dark reaction. That's all. These two you will be learning in today's class. Okay. So let's get into it now. So all of you know what is pigment. In your NCERT, they have given you one small exercise or small experiment called as paper chromatography. Thin paper chromatography. This is the paper chromatography. What do you do, students, in paper chromatography? You take crushed leaves. You take a leaf, completely crush it with the help of a solvent like acetone or alcohol. Okay, acetone you can add, and you strain it with the help of a filter paper, and then we get the extract. Here. Now you need to take the extract. And add drop wise into a paper chromatography. This is called as chromatographic paper. Chromatographic paper, special paper. Okay, not your normal classmate book paper. Okay, it is not classmate book paper, it's special paper. Okay, expend more money for this. Okay, now you take this paper and you add it into a test tube. In this test tube there is solvent. In this test tube. Okay, in this test tube there is a solvent. Can you see this? Here is the solvent is here in the bottom. Some type of solvent. Okay. Now what is happening is this paper can absorb the solvent. This paper right here can absorb the solvent. So this paper is absorbing the solvent. Yes. Solvent is moving up the paper. Now as the solvent is moving up, some amount of extract, whatever is present in the extract is completely solubilizing in the solvent. Yes. So as the solvent is going up, whichever component of the extract is more soluble, I repeat again, whichever component of the extract is more soluble will obviously move up, which is less soluble will be at the bottom. This is how you obtain the paper chromatography. The more solvent will come on, soluble will come on top, least soluble will be at the bottom. And with the help of paper chromatography, you can separate out the components of the plant extract. This is how you obtain the different types of pigments. We have carotenoids, xanthophylls, chlorophyll A and chlorophyll B. all the different types of pigments completely separated based on the solubility in the solvent with the help of paper chromatography. With the help of paper chromatography. This is called as paper chromatography. That's how you obtain the different types of pigments. And all of you know what is a pigment now? All of you know how a pigment functions. A pigment is a chemical compound which can excite the electrons, and convert light energy into chemical energy. You read now. Looking at the plants, you have ever wondered why and how there are so many shades of green in leaves, even in the same plant? We can look for answers to this question by trying out a separation of leaf pigment of any green plant through paper chromatography. A chromatographic separation of leaf pigments shows that the color that we see in the leaf is not due to a single pigment. Obviously, there is no single pigment. There are multiple pigments, right? But due to 4 pigments: chlorophyll A, chlorophyll B, xanthophyll, and carotenoids, as well as carotenoids. Now you might ask me, sir, is this very important to learn the color along with it? Yes, it is very important because it was a previous year question, NEET question. What is the color of each pigment? What is the color of each pigment? And the trick is very simple. This is 𝐂𝐤 𝐎𝐤 right here. So first one is chlorophyll A, that is bright or blue-green in color. Blue-green right here. Blue-green right here is your chlorophyll A. Next one is what? Tell me. Green-yellow. Chlorophyll A is yellow-green. Can you see? Green-yellow is second one. So can I tell chlorophyll B is green-yellow? Next one. Then we have xanthophyll, which is yellow. This is xanthophyll. Okay, yellow. Last one is carotenoids, that is yellow to yellow-orange. Yellow to yellow-orange is going to be carotenoids. That's how you can remember, friends. That's how you can remember the colors of the different pigments, which has been asked previously in your examination. They can ask you again, so be prepared. Xanthophyll yellow, blue-green chlorophyll A, green-yellow chlorophyll B, yellow-orange is carotenoids. Okay. Now, students, let's understand next concept now. Let's understand next concept right now. Let's understand the next concept right now. Okay. Now, in this pigments, students, all of these pigments can be classified into 2 separate categories. All these pigments, students, all these pigments can be classified into 2 separate categories. All these pigments can be classified into 2 different categories. Pigments. The first one is called as chief pigment. Other pigments are called as accessory pigments. Accessory pigments. Chief pigment. Now, I'll give you a simple example. Understand. When you're wearing a nice dress, when you're wearing a nice clothes, for example, if I'm wearing this shirt right now, this is my main clothing. Do you add some accessory to this clothing, like a watch, a chain, sometimes a ring, a glasses, a hat? Some of you add. I don't add, obviously. I look like stupid then if I add it and come in front of class. But have you seen people add? Right? Models, you have seen this supermodels and all. They will have this bracelet on one hand, watch in one hand, 1, 2, 3 bangles, right? Nice gold blank bangles, one nice glasses, one hat, right? One nice shoes. All of this becomes accessories. This is the main dress. So in the case of pigment also, we have a main pigment and certain accessory pigments. The main pigment, students, the main pigment we have here is the chlorophyll A. Chlorophyll A is the main pigment. Chlorophyll A is the main pigment. Then what are the accessory pigments then? The accessory pigments are the chlorophyll B, xanthophylls and carotenoids. As well as carotenoids. All of them are the accessory pigments. All of them are accessory pigments. Now sir, what is the difference between them? How can we distinguish them, and is there any specific role to them? Yes, it is. Because when you learn something called as light harvesting complex, I'll teach you now only. What is the main difference. Now, I'll give you a simple example again. Simple example. Okay, simple example. In your school, have you ever heard this one line telling, teachers will tell, in school teachers will tell, they told me also many times, I'll give you TC. Have they told you? Tell me, yes or no, check quickly. Teachers, have they told you in your school, have they told you, I'll give you TC today, TC. If you jump in the class, if you make a lot of noise in the class, if you don't do your homework, basically anything which will annoy the teacher, they'll tell, I'll give you TC today, TC. In my time, they used to tell, when I was in school, they used to tell, I'll give you TC today. Right? They'll tell, what is the meaning of this TC? Right? What is this TC? TC is nothing but called as transfer certificate. It is a certificate. Be happy. It is called as transfer certificate. With the help of transfer certificate, you can go to next college, next school, annoy there again, take one more TC, go somewhere else. Take one more TC, go somewhere else. Basically, it is called as transfer certificate, and all of you, when you graduate your class 12, you need TC for sure. Everyone needs the TC, okay? TC is very important. Everyone should get the TC, okay? Now, tell me in the school, will the teacher give you TC or will the principal give you the TC? Have you heard in the school? I have gone to principal's office at least 2 times. They used to tell, go to principal's office, principal sir or ma'am will give you the TC. Ma'am or sir, teacher will never give you the TC. Principal will give you the TC. Have they ever happened to you? Tell me. You will never get the TC. The principal sir or the ma'am will give you the TC. Right? Yes. Principal sir or ma'am will give you the TC. Now, what does it tell you? Teachers cannot give you the TC. Principal can give you the TC. So here, students, understand. In the case of plants, also they also have a similar. Plants also have a similar thing. There is something called as reaction center. Reaction center can do all the work. Like how principal can give you CT, reaction center, which is also called as the chief pigment. This chief pigment or the reaction center has the ability to produce the TC or to make the enzymes. It has the enzymes and it can give the reaction. It can excite the electrons. What about axillary pigments? Axillary pigments do not have the enzymes. They do not have the enzymes. All the axillary pigments can do is they can collect the energy and give it to the reaction center. So your chief pigment is called as reaction center, also called as the principal. Axillary pigment is like your teachers. Teacher will come and tell your principal, this person in my class, 10th B or 12th B class student is making a lot of noise. Principal, please take care of this. So axillary pigments like teacher, they'll give all the information, all the light to the chief pigment. Okay, understand now, understand. And 90% times you will get TC because of your friends only, I'm very sure. Because of your stupid friends only, you will go to principal's office 90% of time. 90% of time. That person will make a lot of noise, you will also get stuck. Since they are your best friends, you cannot even tell anything. Both of you take TC and go away. Okay. Now understand. Next concept. Let's read what is given here. Let's read what is given here. The pigments are substances. The pigments are substances that have an ability to absorb light. I told you they can absorb the light. Pigments at a specific wavelength, right? They can absorb the light. A pigment can absorb the light at a fixed wavelength, and it can excite the electrons. Yes, yes. Let us study a graph showing the ability of chlorophyll A pigment to absorb light in different wavelengths. That is figure 11.3. Understand now. Okay, understand 11.3. Where is 11.3? This one right here is the 11.3, no? 11.3 graph. So this 11.3 graph is showing the different absorption peaks of different pigments. Absorption peak of different types of pigments it's showing you. Now, can I teach you absorption spectrum now? Absorption spectrum. Absorption spectrum is very simple. Absorption spectrum is telling you that at which wavelength, which pigment is getting absorbed. Very simple. At which wavelength here? At which wavelength, which pigment is getting absorbed. Is getting absorbed. Can you see here? At this wavelength right here, chlorophyll A is getting absorbed here and it is getting absorbed here. That is chlorophyll A. Chlorophyll A is getting absorbed at two different wavelengths. For example, here we have chlorophyll A is getting absorbed at 420 and 680, I guess. Chlorophyll A 450 and 680. 450 and 680 here. This is the wavelength of light. Chlorophyll A. Similarly, chlorophyll B also be getting absorbed somewhere. Yes, this is around 480 and this will be around 650 before this, 650, right? In these two regions, wavelength is being absorbed. Yes, and these two regions happen to be the blue region and the red region. Here we have the blue region, here we have the red region. So what are we learning here? We learned here that at a particular wavelength of light, a pigment is getting absorbed. This right here is the action spectrum. Action spectrum. Tell me, what is the action spectrum? Tell me, students, what is the action spectrum? Action spectrum tells you at which wavelength work is done. What is the work? Photosynthesis. Work is nothing but the photosynthesis. So at these two regions right here, the blue region and the red region. In the blue region and the red region, more photosynthesis taking place. Didn't we learn this in T.W. Engelmann experiment? In T.W. Engelmann experiment, we learned that in the blue region and the red region, there is increase in photosynthesis. This is nothing but the action spectrum. Now, students, can we merge both of them now? Can we merge the absorption spectrum and action spectrum now? Can we overlap now? Yes, we can overlap because there is some similarity. Can you see? In this also there is a peak here. Here also there is a peak. Here also a peak. Here also is a peak. Now we will overlap both of them. For chlorophyll A. This is for chlorophyll A. Now can you notice one simple thing here now? Whenever in chlorophyll A, at here and at here, that is the blue region and the red region. In the blue region and the red region, there is more absorption. Can you see? In these two regions right here, there is more absorption of light. If there is more absorption of light, can I tell there is more photosynthesis? Yes. There is more absorption and there is more photosynthesis here. In these two regions right here. Yes, sir. Now, students, You tell me in the chat right now. Is it completely overlapping? Tell me, is it completely overlapping now? Is the graph 1 and graph 2, are they overlapping completely? Tell me. Yes or no? Mani, are they overlapping completely? Tell me. This is for chlorophyll A. Both the graph are chlorophyll for chlorophyll A. Are they overlapping completely? No. Can you see right now, students? Can you, all of you, concept here. In this region between 500 to almost 600, in this 500 to 600 range, in this range right here, there is no absorption. Can you notice? In this 500 to 600 range, there is no absorption of light here. There is no absorption of light here. Even though there is no absorption of light in between 500 to 600, there is no absorption of light, but there is some amount of photosynthesis. Even though there is no absorption of light here, we can see some amount of photosynthesis still taking place. Now you tell me, all of you, in your school, if the principal is absent, will the school be closed? Tell me, this chlorophyll A is the chief pigment. Is the chief pigment. Chlorophyll A is a chief pigment. Even though the chief pigment is not absorbed here, it means that the principal is not present. Even if the principal is not present, will the school go on? Yes, the school will go on. School will go on. Right. If the principal is absent that day, will the teachers come to the class and take your class? Yes, they will come and take. Who are the teachers here, students? Who are the teachers here? The teachers here are the axillary pigments. So understand the concept right now. Beautiful concept here. So even though the chief pigment is not absorbed, that is, even though chlorophyll A is not absorbed between 500 to 600, it is not being absorbed. But in that region, 500 to 600 region, can I tell chlorophyll B, some amount of xanthophyll, some amount of carotenoids are being absorbed? Yes, they are being absorbed. Axillary pigments are getting absorbed. And remind me, what was the function of axillary pigments? The function of axillary pigments was to provide the energy to the chief pigment. Obviously, if chlorophyll A is not absorbing the light, chlorophyll B, xanthophyll and carotenoids, those three pigments will absorb the light and they'll give the energy to chlorophyll A. Even though principal is absent, teachers will teach the class, teachers will report to the principal. Teachers will report to the principal telling that class was fine today, no more TC today. No more TC today. Did we learn this concept somewhere, students? Did we learn this concept somewhere? Right here. Transfer of energy to another pigment. So, axillary pigments are excited. Axillary pigments are, light is being excited. Energy is coming down. What is happening here? Transfer of energy to another pigment, that is, your axillary pigments are transferring the energy to the chief pigment. Transferring the energy to the chief pigment. Understood, all of you? Beautiful concept, right? Beautiful concept. It is given in your NCERT. It is given in your NCERT. Understand again, I'll tell. In this region right here, between 500 to 600 region, in between 500 to 600 region, there is no chlorophyll A absorption. There is no chlorophyll A absorption at all. Not happening. That is, principal is absent. Chief pigment is absent here. Chief pigment is absent here. Okay. So even though principal or the chief pigment is absent, your axillary pigments, chlorophyll B, xanthophyll and carotenoids, will they be functional? Yes, sir, they will be functional. So this xanthophyll, carotenoids and chlorophyll B, since they cannot do the work on their own, they will provide the energy. They will provide the energy to the chief pigment. So that's why That is why, students, that is why even though the light blue line is low, even though there is no absorption here, some amount of photosynthesis is happening. Photosynthesis is happening even in this low region. Can you see? That is why this dark black line is not at the bottom, it is somewhere up. Because this black line represents, photosynthesis, action spectrum. That is, photosynthesis is happening or not. Understood, all of you? I thought this concept here also. When we were learning pigments, transfer of energy to another pigment. So what is happening? Axillary pigments are transferring the energy. Axillary pigments are transferring the energy to the chlorophyll A. There is no absorption of light here by chlorophyll A, but work is done by the chief pigment. Okay? Understand now. Let me teach you one more time. Understand this beautiful concept. For example, we have very simple to understand, very simple to understand the entire process. Just listen to me. Okay. Here we have carotenoids. Wavelength is between 500 to 600. Wavelength is between 500 to 600 or 500 to 550. Here we have xanthophylls. Between, that is, 550 to 600. 550 to 600. Then we have chlorophyll A. Then we have the chlorophyll B, which works at 600 nanometer. All this is nanometer. Okay. Now here we have the main boss, chlorophyll A, which works at around 680 nanometer. Understand? 680 nanometer. Understand? Now, the sunlight is coming, right? The sunlight is entering the chart. Sunlight is entering. Now tell me, in the sunlight, we have something called as PAR. Photosynthetically active radiation. Which is between 400 to 700. Now tell me everyone in the chat, can the chlorophyll A absorb the wavelength of 500? No. Chlorophyll A cannot absorb the light of 500. So this range of wavelength of light, some amount of wavelength between 500 to 550 will be absorbed by the carotenoids. Yes, they'll absorb the light. Here. Your xanthophyll will absorb the light from between 550 to 600. They'll absorb the light. Chlorophyll will be absorbing light between 600 wavelength. Now tell me, all of these are your axillary pigments. All of them are axillary pigments, that is chlorophyll B, xanthophyll and carotenoids. Now they cannot work on their own. Like how a principal cannot work on their own, how the teachers cannot work on their own. Teachers have to report to principal. So all the energy which is trapped by the axillary pigments, they will transfer all the energy, students. Listen to me very carefully. They will transfer all the energy to the chlorophyll A. They will transfer all the energy to the chlorophyll A. Understanding? Transfer energy. So that is why students, in this region right here, even though there is no absorption by chlorophyll A, there will be work done by chlorophyll A. Understood, all of you? Deepika, where does the photosynthesis take place? photosynthesis takes place in the red region, the blue region. For that, you need to understand the T.W. Engelmann experiment. So watch the T.W. Engelmann experiment. The concept will be clear to all of you. Okay. Do all of you understand? This part, okay, this one is called as chlorophyll A is also called as reaction center. Reaction center. Because all the reaction takes place there. All the reaction takes place in the reaction center. Okay. All the reaction takes place there. And can you tell all of these chlorophyll B, xanthophyll, carotenoids, they are collectively called as antenna. They are called as what? Antenna. Auxiliary pigments are also called as antenna. The chlorophyll A, the main principle, is also called as reaction center. It is called as reaction center. Reaction center has the enzymes NADP reductase. It has the enzymes and it has the it has the enzymes and it has the ability to excite electrons. All of these following auxiliary pigments will only transfer the energy to the chlorophyll A. Okay. The graph together show that the most of the photosynthesis takes place in the blue and the red regions. All of you know that. Of the spectrum, some photosynthesis does takes place in the other wavelength of the visible spectrum. Let us see how this happens. Okay. How does the other wavelength photosynthesis takes place? All of you know this already. Though chlorophyll is the major pigment responsible for the trapping of light, other thalloid pigments like chlorophyll B, xanthophyll and carotenoids, which are called as the auxiliary pigments, also absorb light and transfer the energy to the chlorophyll A. Indeed, they not only enable wide range of wavelength of incoming light. Obviously, students, if there was only chlorophyll A, then wavelength of light absorption will be limited to only chlorophyll A. Then, since we have multiple auxiliary pigments like antennas, like your 600 nanometer, xanthophyll and carotenoids, can I tell the wavelength of absorption is increased now? For example, if you only have one radio tower, will the signal be stronger? Or if you have multiple radio towers, the signal will be stronger. Multiple radio towers. So we have multiple auxiliary pigments which are increasing the surge of wavelength. See, to be utilized for photosynthesis, but also very important line, but also protect the chlorophyll A from photooxidation. What is the meaning here? Imagine, students, if all the wavelength of light went directly to the chlorophyll A, it would be difficult for the. Imagine if there were no teachers in the school, only one principal. The principal has to do all the work, teach every single class. Principal will go mental then. Yes, obviously. Since the principal has a lot of teachers can go teach everywhere. They can make sure every single class is keeping quiet. Principal doesn't have to go to every class and tell be quiet, be quiet, be quiet. That is called as preventing the photooxidation and increasing the wavelength of absorption. Okay. Clear, students? That is these two lines here are very important. Very important lines of NCERT. Many questions have come from here. Okay. Now, students, before we go to the light reaction, can we take a small 15 minutes break or 20 minutes break? Let's take a 15 minutes break and then we'll come back. Okay. What is the time right now? Time right now is 7:56. So let's come back at 8:10. Yes. Resume the class at 8:10. Resume at 8:10 p.m. Yes. That's a 20 minutes break only because after also relax a little, no? I'm starting from 2 hours. 8:15. Okay. 8:15 p.m. Okay. So we'll start the class at 8:15. Arams will start at 8:15. Go eat your dinner a little bit, have some water, and come back again. So once you come back again, we have to learn what is called as light reaction. What is cyclic and non-cyclic? Cyclic and non-cyclic we'll learn. Then we will also learn something called as PYQs. You see, we have a lot of PYQs. Lots and lots of PYQs to go after that. Lots and lots of PYQs we'll do after that. Okay, lots and lots of PYQs will go after that. So let's start the class at 8:15. Okay. Chalo, I'll see you at 8:15 directly. We'll finish the rest of the today's class there. And trust me, students, learn today's class line by line of NCERT. You will be able to solve all the questions regarding your light reactions. That is my guarantee after today's class. Okay. Session is blue. Yes. Friends, welcome back. I hope your break is over and you were able to, rejuvenate yourself and you're able to, relax a little, and then your brain cells are more activated, because now we're coming towards the end of the session, right? Now I'll be teaching something called as light reaction. After the light reaction, then we'll do questions. Okay. Probably the class will go for next 45 minutes. So stick with me. Okay. So let's go one by one. Let's read what is given here. Before I read what is given here, I want you to understand what is the meaning of light harvesting complex. I want everyone to understand what is the meaning of light harvesting complex. Light harvesting complex is made up of two things. Light harvesting complex is made up of two main things. The two main things are nothing but the reaction center. One second. And antenna, as well as your antenna, as well as the antenna, This is nothing but your LHC, light harvesting complex. It is able to harvest the light. Like we learned here, right? All of you students, come back here. We learned here that LHC is able to harvest the light. Antenna is able to harvest the light and give it to chlorophyll A. That is the reaction center. So here LHC, light harvesting complex, is nothing but reaction center plus the antenna. Together they make up the LHC, light harvesting complex. Now immediately you have a doubt in your mind. Sir, is LHC same as a photosystem? Are they same, or is there any difference between LHC and photosystem? See, this is called as light harvesting complex. Now what is this PS or photosystem? Understand, it's very simple. Again, very simple. Photosystem is made up of three things, actually. Three main things. Photosystem is made up of three main things. Obviously the first one is going to be LHC. It is made up of LHC, light harvesting complex. The LHC is made up of your two things, that is basically the antenna and reaction center. Yes, reaction center. Now apart from this LHC, it is also made up of certain electron carriers such as your pure fighting, PQ, CY, right? All of them are basically type of your electron carriers are present in the photosystem. Apart from electron carriers, there are a lot of enzymes also present here. Enzymes are also present in the case of your photosystem. Enzymes are basically what? Enzymes are basically proteins, right? They're basically proteins. So proteins, electron carriers and light harvesting complex, all three together make up the photosystem. I hope that point is clear to all of you. Students, all this concept your NCERT never tells you such easy manner. It is given in such a complex manner. If you read it once, you will never understand it. Let me show you what is given in NCERT. Reaction center or photochemical phase, all of you know that, includes light absorption. That also is very important. What happens in light reaction? Absorption of light. Light absorption is happening. Light absorption is happening. Apart from that, water is splitting. Water is being split here. Apart from that, what is happening? Oxygen is being released. Yes, oxygen is released. And last one is formation of ATP and NADPH. All of this formation happens in the case of your light reaction. In a light reaction, what is happening? Light absorption, water splitting, O2 is being released and ATP and NADPH are being formed. Remember those points very clearly. They can ask you a question from there directly. Okay. Now, let's read lines of NCERT. Okay. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes. Their group pigments are made into what? Pigments are made into light harvesting complexes, where within the photosystem 1 and photosystem 2. So your light harvesting complex can be found within the photosystem. Light harvesting complex, they are found within the photosystem. Okay, we have photosystem 1 and photosystem 2. PS1 and PS2. PS1 works at 700 nanometer. PS2 works at 680 nanometer. Okay. Now, why is 700 called as first? Because PS1 was discovered first. That's all. These are named in a sequence of the discovery and not in the sequence in which they function. For example, students, your photosystem 1, PS1 works at 700 nanometer and your PS2 works at 680 nanometer. 1 700, 2 680. Not the way they function, based on the discovery. Today, not the entire chapter. Today we will stop the class with entire light reaction, cyclic and non-cyclic. Because I'm going slow, students understand the lines of NCERT. See, I can teach the entire chapter in 3 hours. The entire chapter can get over in 3 hours, but where is the fun in that, right? Do you not want to study the entire detailed lines of NCERT like this, right? All of you want to learn, right? So if you want to learn lines of NCERT, it will take more time. And I cannot do this line by line NCERT when the exam is 2 months away or 1 month away. That time we will do revision on the channel. When the exam is closer to you, we will do revision that time. That time there will be no time for detailed study. All of you understand that, right? So do the detailed study right now. You have more than around 240 days. Right? You have 240 days. So use that time right now. Okay. Now, these are named in sequence of their discovery, not in the sequence in which they function. Okay, not in the sequence they function. During the light reaction, the LHC are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system, also called as antenna. So antenna will not have chlorophyll A. Antenna will be made up of the other. Antenna made up of axillary pigments. Chlorophyll A is the reaction center. Okay. Now, these pigments help to make the photosynthesis more effective by absorbing the different wavelength of light. A single chlorophyll A molecule forms the reaction center. All of you know reaction center. Okay. The reaction center is different in both photosystems. For example, in the case of chlorophyll A, so in the case of photosystem 1, the reaction center works at 700 nanometer. In photosystem 2, it works at 680 nanometer. See, photosystem 1, the reaction center chlorophyll A has an absorption peak at 700. Hence, call it P700. PS2, P680. We learned here. 1 at 700, 2 at 680 nanometer. Okay. Now, students, did we learn this, Did we learn this today only? All of these are axillary pigments. What is this? Here we have a reaction center. This right here is the chlorophyll A. All the pigment molecules, the accessory pigment molecules will take the energy and give it to the reaction center, that is the chlorophyll A. All the energy is given to the chlorophyll A. Chlorophyll A, excitation of electrons. Primary acceptor, which I'll teach you. Tomorrow's class, don't worry. Tomorrow there is no class. The next class is this week itself. This week you have 3 classes, students. This week you have 3 different classes. Actually 4 classes. We have increased the number of classes for you. This week I'll be teaching you line by line of part 2 of this particular chapter. Then I'll be teaching you line by line of biomolecules. Yes, line by line of biomolecules. 2 parts in this week. Okay, 4 classes. 4 classes this week. So what we learned till now today? Photosynthesis happens in 2 phases. We have the light reaction, we have the dark reaction, also called as light independent reaction. Light reaction occurs in the chloroplast. Trapping of light, ATP is produced. This ATP and NADPH is used in the dark reaction. Synthesis of sugar by the utilizing the ATP and NADPH happens in the dark reaction. Okay. Now students, now we come to the light reaction, the main topic for today. Okay. The entire light reaction is divided into 2 parts. Light reaction itself is divided into 2 parts. We learned what is LHC, we learned what is photosystem. Now we use those concepts in the particular, this concept right here. Here we have something called as non-cyclic photophosphorylation. Then we have something called as cyclic photophosphorylation. Phosphorylation. What is the meaning of this? The meaning of photo, light, phosphorylation is formation of ATP. Using the light energy, ATP is being produced in a cyclic manner. Using the light energy in a non-cyclic fashion, ATP is being produced. Hello, Dr. Gumandi Srinivas. Okay. Hi to you. Okay. So students, now we will learn something called as cyclic and non-cyclic. Ready? Ready, students? Ready? Tell me in the chat quickly. Ready, all of you? Now we will learn something called as cyclic and non-cyclic. Understand every single step from this point onwards for the next 20 minutes. It's going to be very crucial. If you want to solve those difficult questions, it will come from here. So focus right now if you want to solve this question in the examination. All of you. Ultimate focus now. Go to ultra instant. Okay. First we will learn something called as non-cyclic. First we will learn the non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Okay. Ready? Will you tell me in the chat, ready all of you? I want some Josh in the chat. Okay. So we'll start from here and then we'll go towards that side. Okay. We will start from this side and we'll move towards the other side. Okay. Now tell me the reaction center now. First of all, reaction center. In the non-cyclic students, in the case of non-cyclic, we have one reaction center here. And I will call this reaction center as chlorophyll A. Yes, chlorophyll A. And this reaction center is working at 680 nanometer. It is working at 680 nanometer. Now tell me, if it is working at 680 nanometer, is it photosystem 2 or 1? See, 1 at 700, 2 at 680. So this is your PS2. PS2 working at 680 nanometer. Now, if this is the reaction center, will it have auxiliary pigments? Yes. So can I tell we have the auxiliary pigments here? Auxiliary pigments around it. Okay. Now we have sunlight coming in. Sunlight is coming. Sunlight is falling. Okay. All the auxiliary pigments will send the energy to chlorophyll A. Yes or no? Light harvesting complexes. They'll send the energy to chlorophyll A. Everyone is sending the energy. Different color will use. All the energy is being sent to the chlorophyll A. Okay. All the energy trapped by the light, sunlight, which is in your PAR, photosynthetically active radiation. it is taken up by auxiliary pigments also. Energy is being sent to chlorophyll A. Now, from this chlorophyll A right here, electrons is being emitted. Yes. Electrons are being emitted. Now, this electron is taken up by a primary electron acceptor. It is taken up by something called as primary electron acceptor. In the case of non-cyclic photophosphorylation, the primary electron acceptor is pheophytin. It is going to be pheophytin. Okay, pheophytin is taking up the electrons. Okay, pheophytin is the primary electron acceptor in the case of photosystem 2. Okay, from here electrons are being sent. PQ, CY, PQ, CY and PC. PQ, CY and PC. Now you won't be wondering, sir, what is this PQ, PY, PC now? PQ, CY and PC. Become too low. I need space that side. CY. Electrons are being sent. Okay. Now you have to understand what is this PQ, CY, PC first of all. What is this? And there is a small box here only. I'll write down. What is this? PQ, CY and PC. Understand? I'll write here only for you. Then you can refer to it again back. Okay. Here, first of all, PQ is called as plastoquinone. What is CY? CY is nothing but cytochrome B6F complex. B6F complex. What is PC? Priyanka Chopra? No. Plastocyanin. That is PC. Okay. Now from PC, the electrons are being transferred. Electrons are being transferred from PC. Okay. From PC here, the electrons will be taken up by the second chlorophyll reaction center or the second photosystem. Again we have chlorophyll A. Again we have the chlorophyll A here. Again, do we have the different types of auxiliary pigments here also? Auxiliary pigments are also here. Will this also receive light separately? Yes, it will also receive light. Okay. Now this particular system is working at 700. Can I call this photosystem 1? Yes, can I call this photosystem 1? Yes, this is photosystem 1. Photosystem 1 working at 700 nanometer. Photosystem 2 working at 680 nanometer. Again, all the energy will be given to this auxiliary pigments. Auxiliary pigments is getting the energy. Okay. Now again from here, the electrons will be emitted out. Electrons are being emitted out. Again, the electrons will be taken up by the primary electron acceptor. Primary electron acceptor. Yes. Now here, in case of your PS2, the primary electron acceptor was pheophytin. It was pheophytin. But in the case of your chlorophyll 2, the primary electron acceptor is going to be iron sulfur centers. Iron sulfur centers. Iron sulfur centers are going to be primary electron acceptors. Okay. Now from here, the electrons will send to FD. Electrons will be sent to NAD plus. NAD plus. Okay. Let's draw that. Okay. Electrons being sent here. Being sent here. 2 electrons. Okay. FD is ferredoxin. FD is ferredoxin. Now here what happens is this NAD plus. Tell me students, starting of the class I told you. In the starting of the class I told you. In the starting of the class I told you. Taking in electrons and also addition of protons is called as what? This proton right here is coming from the stroma. This proton right here is coming from the stroma, understand that. Addition of proton and addition of electron is called as reduction. So this NAD plus, which is the final electron acceptor here, undergoes reduction. It undergoes reduction to make NADPH2. Becomes NADPH2. NADPH2. And this entire process here is mediated by an enzyme called as FNR enzyme. Ferredoxin NADPH reductase. Ferredoxin NADPH reductase, that is the enzyme here. Enzyme, FNR enzyme. FNR enzyme. So what did we learn right now? What did we learn right now here is chlorophyll, your PS2 is absorbing the light at 60 nanometer, electrons getting excited, primary electron acceptor pheophytin, PQ, CY, PC, plastoquinone, cytochrome BCX complex, and plastocyanin. Electrons are then taken up by PS1, 700 nanometer, again electrons are getting excited, primary electron acceptor is FES, then FD, then final electron acceptor. The final electron acceptor is NAD plus, yes, NAD plus. Taking up electron and converting it to NADPH2. NADPH2, it is getting reduced. It is getting reduced here. And the proton is coming from stroma. FNR is the enzyme. Ferredoxin NADPH reductase. NADPH reductase is the enzyme here. Okay, all of you agree with me? Now students, I wanted to understand, if electrons is being consumed here, how are we getting more and more electrons? Tell me. If electrons are being consumed here, how do we get ultimate source of electrons? Tell me. How do we get ultimate source of electrons? Electrons are being excited here. They are lost there. They are being reduced. So how do we get continuous source of electrons? The continuous source of electrons is given by the water actually. Did you remember students, we learned in the starting of the class, water is the hydrogen donor. It's also the electron donor here. Okay. So here we have observed in the inner lumen. In the inner lumen we observed there is photolysis of water. The water is being split. Water is being split into 2H plus oxygen and the electrons. This 2H plus right here, this 2H plus light right here will be used in the formation of ATP. This water is the byproduct here, and this electron will be taken into the cycle. In the inner thylakoid membrane, this is happening. This all is happening in the inner thylakoid membrane. Now students, what did we learn from this entire thing? What did we learn from the entire experiment? Did we learn that, students? Did we learn that? First of all, can I tell oxygen is being evolved here? First point, O2 is evolved. Just right. O2 is evolved. Yes, O2 is evolved here. First thing, what is being evolved? Can I tell photolysis of water? Photolysis of water. Photolysis of water. This photolysis of water here. Okay. Now, do you see any ATP formation anywhere? Do you see ATP formation anywhere here? Yes, there is, but I'm not mentioned right now. Between your PQ and CY, using the electron, one ADP gets converted to one ATP. One ATP. So can I tell ATP formation? Yes. Is there NADPH formation? Yes. NADPH2 formation. Yes, it is there. NADPH2 formation is happening. Tell me, is it cyclic or non-cyclic now? Is the electrons going back? No. It is non-cyclic in nature. Non-cyclic. Non-cyclic in nature. Non-cyclic in nature. And also, does it look like a Z shape? Z shape? Yes. So can I also call it Z scheme? It's also called as Z scheme. It is also called as Z scheme. Clear? Non-cyclic, and is also called as your Z scheme. Okay. This is what is meant by the entire non-cyclic photophosphorylation. NCERT doesn't tell you in detail, but they will ask you question in detail. I don't know why. They will never tell you this detail, but NCERT will ask you the detail. And students, if you want these notes, you will have to join the Telegram channel. Just remind me once. I am very forgetful sometimes. So remind me, I'll upload the notes. Okay. Clear? No. Now that is non-cyclic done. Non-cyclic done and dusted. Okay. Now how do you understand cyclic now? What is cyclic then? Cyclic is very easy. Cyclic is very easy. Cyclic is going to be very, very easy. Because in the case of cyclic, there is a single photosystem. There is a single photosystem here. There is a single photosystem. Every single photosystem has antennas. There are antennas here. Antennas. There is light is falling here. Light is falling. They're trapping the light. They're trapping the light and sending it to the reaction center. Sending it to the reaction center. The reaction center is again made up of chlorophyll. And this works at 700 nanometer. So can I tell it is PS1 then? Yes, PS1 working at 700 nanometer. Now you tell me, where do you find only and only PS1? All of you? Tell me, where do you find only and only PS1? If you go back to today's class starting, didn't I teach you right here on the stromal lamellar? Do you find only PS1 here and not PS and PS2 both? Only PS1 here? Yes, sir. Only PS1 is found. Only and only PS1 is found there. Okay. So can I tell we find only PS1 here? That is your stromal lamellae. Yes. Okay. Stromal lamellae. We find only PS1 there. Yes. Only PS1 is found there. True. Will the electrons get excited now again? Yes, electrons will get excited again. the primary electron acceptor here. The primary electron acceptor. In the case of cyclic, the primary electron acceptor is none other than the iron sulfur centers. Similarly, we had here, right, iron sulfur centers. Similarly, we have the iron sulfur centers here. From here, it goes to FD. Similarly, ferredoxin. But from ferredoxin, now it is going to PQ, CY, and PC. And PC. That is from ferredoxin to plastoquinone, plastoquinone to cytochrome B6 complex to plastocyanin. From plastocyanin, the electrons are again sent back to the photosystem. Again sent back to the photosystem. This is called as a cyclic photophosphorylation. Cyclic photophosphorylation. Now between FES and FD, here we have one formation of 1 ATP, ADP to ATP, ADP to ATP formation. Between PQ and CY, one more ADP to ATP formation. ATP formation in total of 2 ATPs. In total of 2 ATPs. Now tell me, students, in the case of your cyclic, is there any release of oxygen? No, there is no oxygen at all. There is no oxygen at all. Is there any release of NADPH? No NADPH at all. Is there photolysis of water? There is no photolysis of water. Is it non-cyclic? No, it is non-cyclic. It is completely cyclic in nature. It is completely cyclic in nature. This is called as cyclic photophosphorylation. Cyclic photophosphorylation. Okay. Done. Let's read it today, students. We will do the questions in the next class only. That's better. Otherwise, it will take such a long time. Okay. Let's see what we have learned, students, till now. Let's see what we have learned. The photosystem 2, the reaction center, the chlorophyll A absorbs 680 nanometer wavelength of red light, causing the electrons to become excited and jump into an orbital further from the atomic nucleus. These electrons are picked by the electron acceptor, which pass them to the electron transport. One after another, they'll keep on. Okay. Here, the final electron acceptor is again back to chlorophyll A. Starting from here, ending from here. Okay. No. The electron transport system consists of cytochromes. This movement of electrons is downhill in terms of oxidation, reduction, or redox potential scale. The meaning of redox potential scale is very simple. That is, ability of a compound or ability of a particular element to absorb the electrons. Okay. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of photosystem 1 in non-cyclic. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive the red light from the wavelength of 700. I told you, they also get excited on their own. They also receive light, electrons get excited here. Okay, electrons also get excited. Okay, and transfer to the another acceptor molecule which has a greater redox potential. That is, an acceptor which can accept electrons better. Okay, final electron acceptor in the case of your cyclic is going to be chlorophyll A only. Coming back, shoot it out, all of you. Okay, the electrons are then moved downhill again. This time, molecule of energy rich NAD plus, the final electron acceptor, which reduces to NAD plus to NADPH2. This whole scheme of transfer of electrons starting from PS2, first is the uphill to the acceptor, downhill to the electron transport chain to PS1, excitation of electrons transfer to the another acceptor, and finally downhill again to NAD plus. That is, students, first we have the uphill, downhill, acceptor, uphill, downhill again, downhill, again the downhill. Okay, downhill, NAD plus reducing NAD plus is called as Z scheme. It is called as your dead scheme. It's called as Z scheme due to this characteristic shape. Okay, this shape is formed when all the carriers are placed in a sequence on the redox potential scale. That is called as the entire Z scheme. Entire Z scheme. Okay, I hope you understood what is Z scheme now. Again, this is what we learned, right? Light harvesting complex, that is basically your reaction center. It's very simple. What is happening in uphill? You can look at the diagram and tell. See, in uphill, what is happening? In the uphill, electrons are getting excited. In the downhill, PQ, PC. Again, uphill, electrons are getting accepted. Again, downhill, FD, NAD plus is the final electron acceptor. Final electron acceptor. Okay. See here, electron are getting accepted. The first electron acceptor. Yes. Photosystem 2 is first. Then electron transport system we learned. PQ, PC we learned, right? PQ, PC one by one we learned. It's going down. Again, this is your PS1. Electron acceptor. Finally, it is going to NAD plus is getting converted to NADPH. Here, photolysis of water is happening. You see, photolysis of water is happening. One by one, the entire diagram we decoded. Okay. This is the splitting of water which is telling. See, water splitting is happening here. That is, how does the PS2 supply electron continuously? The electrons that were moved from the PS2 must be replaced. Obviously, this is achieved by electron availability due to the splitting of water. The splitting of water is associated with PS2. Told you, associated with PS2, right? It is associated with PS2, splitting of water here, right? Splitting of water into hydrogen, oxygen, and electron. This create oxygen, one of the net products of your photosynthesis, which is removed out later on. The electrons need to replace those removed photosystem 1 are provided by the photosystem 2. So photosystem 1 is using the electrons to reduce NAD plus to NADPH2. Electrons are again sent to the photosystem 1 with the help of splitting of water. We need to emphasize here that the water splitting complex is associated with the PS2, which itself is physically located on the very important inner side of the membrane of thylakoid. Thylakoid is there. This is the thylakoid. On the inner side of thylakoid, can you see this is called as the inner side of thylakoid. On this inner side of thylakoid, we have the splitting of water. This is the entire thylakoid, inner side of the thylakoid. Okay, clear? Then where are the protons and oxygen formed, likely to be released? Proton is lead to the ATP synthesis and oxygen is released outside. Okay. Now, cyclic and non-cyclic. Living organisms have capability of extracting energy from oxidizable substances and store in the form of bond energy, like your ATP carrying the energy to chemical bonds. Okay. ATP is synthesized by cell by mitochondria. All of you know that. Yes. By chloroplast. Is namely photophosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. I told you, using light energy, we are converting making the ATP. When the two photosystems work in a series, first PS2, then PS1, it is called as non-cyclic photophosphorylation. The two photosystems are connected through an electron transport chain. We learned PQ, CY, we learned. Connected through a transport chain and seen earlier in a Z scheme. Both ATP and NADPH are synthesized by the electron flow. So both ATP and NADPH are produced by non-cyclic photophosphorylation. Okay. When only PS1, that is cyclic, the electrons is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. Where this could be happening is in the stroma lamellae. Why only stroma lamellae? Because stroma lamellae has the only PS1 is present. Okay, while the membrane or the lamellae of the grana have both PS1 and PS2. That is thylakoid or grana. Okay, PS2 and the PS2, the stroma lamellae lacks the PS2. I told you, as well as the NADPS reductase enzyme. FNR enzyme is absent in the case of your stroma. In your PS2, the excited electrons do not pass to NAD plus, but is cycled back to PS1 complex. Cyclic, right? Cyclic. Here, cyclic is again sent back to the chlorophyll. Again sent back to chlorophyll, right? Through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP. So only ATP in the case of your cyclic, not NADPH2. The cyclic photophosphorylation also occurs when the only light of wavelength beyond 680. Why? Because chlorophyll A, photosystem 1 works at 700 nanometer. 700 nanometer. Okay. Beyond 680 are available for excitation. See here. Chlorophyll, this is your photosystem 1. Electron is getting excited. Primary electron acceptor, that is your FES, electron transport system. ADP to ATP formation. Again, electrons are cycled back to your chlorophyll 1. Again cycled back. So with that, the first part of the lecture is over. Now you might be wondering, sir, you did this for 3 hours. 3 hours you did this, sir. Students, tell me, did we miss any single point of NCERT now? Did we even miss one point of NCERT which you could not understand? Obviously not. Right. So this is what NCERT line by line means. Even though the lectures are going to be longer, right? It's going to be longer. You will be able to solve all the questions now. You will be able to solve all the questions. Okay. Can we do the PYQs now, students? For next 10-15 minutes, can we do the PYQs? And then we can stop the class. Okay. Ready? Ready for PYQ? First question is here from NEET 2024. Read. The photochemical phase. Tell me, what is the meaning of photochemical phase? What is the meaning of photochemical phase? Tell me. The meaning of photochemical phase is the light reaction or dark reaction. Please tell me in the chat. Photochemical phase is the light reaction or dark reaction? It is the light reaction. Yes. What does it include? Release of oxygen will happen. Formation of NADPH. Is there any use of ATP? No. There is no use of ATP. Okay. Water splitting is happening? Yes, water splitting is happening. Is carboxylation happening? No, it's not happening. Not happening. So what is the answer now? Tell me. What is the answer? A, B, D. Right? A, B, D is the answer. A, B, D is the answer. All of you understood? Light reaction, all of you know. Oxygen is released, ATP is formed, but there is no use of ATP. But is there formation of ATP? Yes, ATP is being formed, but there is no use of ATP. Okay. Next question. The reaction center PS2 has an absorption maxima at, see, very easy question. PS2, so 680 nanometer. 680 nanometer. PS1 700. PS1 700. Next question. Which out of the following statement is incorrect? Incorrect statement. Okay. Grana lamellae has both PS1 and PS2. Grana lamellae, grana has PS1 and PS2. Okay. Cyclic photophosphorylation includes both PS1 and PS2. Obviously not. Cyclic will only have what? Cyclic will only have PS1. Will only have PS1. PS2 will be absent. So our answer is obviously B. Right? Obviously B is the answer. Incorrect one. Incorrect statement. Done. Next question. The products of a light reaction in photosynthesis are light reaction. Do we get ATP? Yes. Do we get NADPH? Yes. Do we get oxygen? Yes. Do we get water here? No. Water splitting is happening. Do we get water again? No. Do we get CO2 release? No. CO2 is not released. CO2 is not released. So answer is obviously first. First is the answer. Very simple. Do you see that? You can solve any question given to you now. Next question. See, in a light reaction, plastoquinone. See, this is a difficult question. In a light reaction, students, when you were reading NCERT, Did we ever read plastoquinone anywhere? When you were reading the NCERT till now, did we ever read the mention of plastoquinone? Did you ever read the mention of cytochrome B6F? Did you read any mention anywhere? Obviously not. There was no mention of plastoquinone. There was no mention of cytochrome B6F complex. It is only shown in a diagram which I'll show you in next class. Next class I'll show you that, where it is written. Okay. Nowhere they have mentioned something called as your cytochrome B6F complex. Nowhere they have mentioned in your NCERT. Only place they have mentioned is in a diagram, which I'll show you later on. Okay. This is what it means to learn from NCERT line by line. Okay. So plastoquinone facilitated the transfer of electrons from. Okay. Let's go back. Plastoquinone. Okay. What is plastoquinone? You have to go back to the diagram. Plastoquinone, I hope you can see in small diagram. Plastoquinone, does it send from cytochrome to PS1? No. Does not send. See, plastoquinone is here, PQ. Does it send from CY to PS1? No. Your plastoquinone is between your PS2 and cytochrome B61. Is it between them? Yes, it is between them. It is between them. Do you understand? What is the answer? Tell me. What is the answer? It is between PS2 and cytochrome B6F complex. PS2 and cytochrome B6F complex. So answer is option number 4. Okay. Now students, here is a big question which you probably might not be able to read. Let me tell you. T.W. Engelmann experiment. T.W. Engelmann experiment with cladophora. Which two different regions of light did he observe higher photosynthesis? Which color? Easy. Blue and red. Blue and red, higher wavelength. This is the NEET 2019 question. Next question. Which of the following is not a product of your light reaction? Not a product of your light reaction. Tell me. Do we get ADP? Yes. NADPH? Yes. Do we get oxygen? Yes. But did you, students, did you see me writing anywhere NADH? Did you see me writing anywhere NADH? No, never. I always wrote NADPH. NADPH. Now I'll tell you a small trick here. In this chapter right here, students, in this chapter right here, you will always encounter NADP. P, phosphate will be here. In the next chapter, respiration in the plants, you will find NADH. Respiration plus NADH. Now you will be like, sir, I want to know the difference. Can you tell me the difference between them? Students, here's the difference. Here's the difference. I'll give you the PPT. You can see the entire difference between your NADPH and NADH. Nicotine adenine dinucleotide phosphate, nicotine adenine dinucleotide. I'll give you the PPT. You can read the entire difference. Very simple. But simple thing you need to understand here is NADP will come in photosynthesis, NADH will come in respiration. That is a simple thing you should need to know. NADH will come in your respiration. NADP will come in your photosynthesis. Simple. Okay, next question. See, all the following regarding the role of xanthophyll and carotenoids in photosynthesis are correct except, tell me which one is wrong. That is actually pigments, yes. They enable wide range of wavelength incoming light, yes. They protect the chlorophyll A from photooxidation, yes. They peak absorption in blue and red, no. Blue and red is for chlorophyll A. Chlorophyll A. Okay, chlorophyll A. So incorrect statement. Next question. In photosynthesis, light dependent reaction takes place in light independent reaction. Light independent reaction. Light independent reaction means what? Dark reaction. And dark reaction happens where, students? The dark reaction happens in the grana. Right? So dark reaction happens in the stroma, the matrix. Stroma. Dark reaction stroma. Right? Stroma. This is a question from NCERT NEET 2015 question. Electrons from excited chlorophyll molecule of photosystem 2 are accepted first by. So this was a controversial question in that year. Primary electron acceptor is pheophytin. Primary electron acceptor is pheophytin. Now you might notice pheophytin is not given here. If pheophytin is not given, which is the next electron acceptor? Tell me. The next electron acceptor after pheophytin is the plastoquinone. Plastoquinone. So answer they gave that year was plastoquinone was the given answer. Plastoquinone was the given answer. Understood? This is called as most probable answer. Plastoquinone. Next question. In photosystem 1, the first electron acceptor. In photosystem 1, the first electron acceptor. In photosystem 1. Photosystem 1 will go up and is given by iron sulfur centers. Yes, iron sulfur centers. Do you understand? So iron sulfur proteins, easy question. You realize if you don't learn what I have given you, students, do you realize I have given you something called as complete chain, cyclic and non-cyclic. If you don't learn the exact thing, you cannot solve such questions. You cannot solve such questions. It's a NEET 2006 question. So this is what it means to learn the plant physiology. And I've made sure you learn every single one of them, every single point. Okay, iron sulfur centers. Next question. The production of NADPH in chloroplast takes place in. Where does it happen? NADPH is produced where? NADPH is produced in your non-cyclic, right? Non-cyclic. In the case of non-cyclic, do you get NADPH in cyclic? No. In case of cyclic photophosphorylation, you will only get ATP. Cyclic only ATP. Non-cyclic NADPH. NADPH. Non-cyclic. Along with NADPH, you also get ATP also in non-cyclic. And this question was from NEET. It was also NEET PYQ, it's not mentioned here. Next question here. The photoautotrophs organisms that do not split water to obtain reducing power are called as. All of you know, right? Such as your H2S in your bacteria, purple sulfur bacteria, right? And green sulfur bacteria. So purple sulfur bacteria is given here. See, this is green plants, now green bacteria. Okay, green plants, purple bacteria. Next question. Which of the following, it's an AIMS question, AIMS 2009 question, AIMS question. Which of the following is a character of your PS1? Of PS1. It's a tricky question, very easy question, but very easy question. It activates only up to 680? No. PS1 can activate up to 700. Reaction center is PS1 is 680? No. Reaction center is P700. PS1 is reduced by electrons released in photolysis of water? No. Photolysis of water will happen only where? In PS2. In PS2. So wrong statement. PS1 is involved in non-cyclic photophosphorylation? Yes. It is involved in non-cyclic also. You remember, right? Non-cyclic has both. Right? PS2 and PS1 both are present. So option number 4, not 3. It's a very simple question, yet little tricky. You might always think that PS1 is associated with cyclic always. PS1 is always with cyclic. No. Remember, students, PS1 is present in your non-cyclic also. Right? All of you understood now? Option number 4. The PS1 is reduced by electron released in photolysis of water? No. Photolysis of water will happen only in the case of your PS2. Beautiful question. AIMS. Again an AIMS question, all of you. AIMS 2010 question. Cyclic photophosphorylation involves. What do you get in cyclic? Very simple question. In cyclic, what do you get? Very simple question. Cyclic, what do you get? PS1, PS2, both PS1, PS2, but only PA680. All of you know cyclic is only what? Cyclic is only PS1. 700 nanometer. Done. Cyclic will have only PS1. Remember, cyclic will have only PS1, not B. Okay. Cyclic will have only PS1. Non-cyclic is going to be PS2 and PS1 both. If this was, students, tell me, if this question was non-cyclic, then what is the answer? If this question was non-cyclic, then what is the answer? Tell me. If this question was non-cyclic, then what would be the answer? If this was non-cyclic, then the answer would have been option number 3 then. Because non-cyclic involves both PS2 and PS1, both are there. Such a beautiful question. Such a beautiful question. Okay. Next question, AIMS 2011 question. PS2 occurs only in. PS2 occurs only in. Where does it happen only in? See, obviously not matrix. Okay. Obviously not stroma. Obviously not stroma. Obviously not stromal lamellae, because we know in stromal lamellae we only have PS1. PS1 only in stromal lamellae. So what is the answer then? Answer is granule thylakoids. Because we know granule thylakoids have both. They have both PS2 as well as PS1. Both are present. Beautiful. Easy question. AIMS 2011 question for all of you. AIMS 2015 question. 2015 question AIMS. Carbon dioxide is necessarily for photosynthesis. The chemical used to remove the gas effectively from the entering the control apparatus was given what? KOH. All of you know. Very simple answer now. Potassium hydroxide solution. KOH will remove all the CO2. Moles half-life experiment. KOH will remove all the CO2. AIMS 2016 question. Which of the following is incorrect about light reaction from the photosystem? P680, P700 are reaction center of PS1 and PS2 respectively. Okay, we'll wait. Light energy provides energy for photolysis of water through excitation of a reaction center at PS2. Yes, PS2 photolysis of water. Makes sense. Okay. NADPH2 is not produced in cyclic electron transport in light reaction. Tell me. Correct? No. Correct. NADPH is not produced in cyclic. NADPH2 is not produced in cyclic. Okay. Reaction center of two photosystems are needed for production of NADPH. Yes, both are required. But what the first one? It says that PS1 at 680, which is wrong. PS1 is at 700. PS2 is at 680. So they have given ulta here. Ulta. NEET 2020 question. This is your homework question now. Students, do you realize how many questions you solved? Homework question. And tell me how was today's class in the comment section today. I will check everyone's comments. That is Sukanya, Hebron, Serotonin, Pahul, Sumi, Roshni, Madhumita, Ravindrani. Everyone's comments I'll check. This is the homework question. That is, during non-cyclic photophosphorylation, when electrons are lost from the reaction center PS2, what is the source of replenished electrons? Very simple question. I want the answer in the comment section. And students, yes, it took a lot of time. Yes, we did a class for more than 3 hours. But do you get that confidence within you right now? Tell me. Do you have the confidence within you that, yes, sir, the concept is so clear right now to me that I can clear this examination? This is what I want from my students. This is what we believe in NCERT line by line. Right? NCERT line by line. Okay? Students, if you want to get into DNA batch or if you get into any of the paid batches for free, there's a trick actually now. There's a trick for getting into paid batches. Any offline batch also. There's an exam called as NVSAT exam. Link in the description. NVSAT exam, link in the description. If you are smart and clever and if you think you can clear the examination, write the NVSAT exam today. Not today, obviously. Enroll for the NVSAT exam. And you can get into Vedantu, paid batches for almost like 90% off. So check out the NVSAT examination. Even if you don't get into full scholarship, you can get iPad, iPhones, drone. So many things are here. So go check it out. All the students who are watching it recorded, smash the like button and also let me know in the comment section if you are mastering the photosynthesis chapter. Okay. So with that being said, thank you so much, students. I hope the energy is going to be up. I hope you are motivated enough to study for the next few days coming up, because long lectures are coming. Long lectures are coming to your way, because we want to teach you in detail and we want you to make sure that you do not skip anything. Okay. So light is the answer. Huh? What light answer? What is this? Light is the answer. Huh? Tell me the answer in the comment section. It is not light and all. Some scam you're doing to me. Tell me the right answer in the comment section. Okay. So with that being said, thank you so much, friends. Bye bye. Take care. Boy, all of you. Let me know in the comment section, all of you. Tell me the answers in the comment section. Okay, I'm checking.